Hi, my name is Emily Chang. I'm a chemistry student working in the Dirksen Research Group under the supervision of Dr. Darren Dirksen. Today, I'll be talking about the research project that I've been working on called the mechanistic study of a novel method of synthesizing a crucial drug scaffold called Azetidine. Scientists have found that approximately 75% of the United States FDA-approved small molecule drugs have nitrogen in them. One of the most prevalent drug scaffold, or core, is the nitrogen-containing molecule called azetidine. As you can see, azetidine is a four-membered ring with a single nitrogen atom. Literature have shown that azetidines provide advantageous properties to drugs, such as improved aqueous solubility, which in turn enhances the drug uptake. Here are some examples of azetidine-containing drugs. We have baricitinib, which is a treatment for rheumatoid arthritis. We also have varenicline, which is an analgesic for acute and chronic pain. And we have dalafloxacin, which is an antibiotic drug that treats bacterial skin infections. There are many more examples that have azetidine in them. There are numerous ways to synthesize azetidine. The two common ways are intermolecular cyclization and intramolecular cyclization. Inter meaning outside or external, and as you can see, azetidine ring is formed from the cyclization of an external compound. Conversely, in intramolecular cyclization, the linear starting material, or substrate, cyclizes on its own and form an internal azetidine ring within the molecule. This sparked interest in the Dirksen group, and the group has proposed a method of synthesizing azetidine through intramolecular cyclization with the use of an iodine catalyst. One of the grad students in our group, Duncan Brownsey, has come up with a set of reaction conditions that made this reaction work. Duncan used PETA as an iodine catalyst, cesium carbonate as a base additive, and TFE as the solvent of the reaction. The reaction took place at 80 degrees Celsius for 16 hours. Duncan was able to get a promising yield of 58% of the azetidine product. This leads to my objective and research question for my project, which is to find out what is the mechanism for this reaction. And if we know the mechanistic information, we could optimize the reaction further and potentially publish this method. In order to answer this question, a series of experimental steps needs to be executed. First, I would have to synthesize a series of substrates that have different substituent or groups attached to it. Then I would perform kinetic experiments on those substrates that I've made. And lastly, I would use the data collected from the kinetic experiments to do computational analysis and generate a plot that could provide mechanistic information about the reaction. In the synthesis step, I've proposed 7 designs of the substrates that I will be making. Each substrate has a different substituents on the right-hand side of the molecule, as highlighted in pink. The substituents are separated into 2 categories: electron donating groups, EDG, which, as the name implies, donates electron density to the structure of the molecule. Electron withdrawing groups, EWG, draws electron density to itself away from the structure of the molecule. The electron withdrawing and donating ability of each substituent is quantified by the respective sigma pair value. These values will later be used to construct a plot for mechanistic interpretation. The different substituents could affect the structure electronically and in turn influence the mechanism and rate of reaction. This is currently where I'm at with the project, and so far, I've made the circled substrates. Once I've made all my substrates, I can move on to the kinetic experiments. During the experiment, I will first add all the components of my target reaction, such as the substrate, iodine catalyst, base additive, and solvent, into a sample tube. Then I will place the sample tube in an instrument called nuclear magnetic resonance, NMR for short, in which the NMR will collect data as the reaction progresses. And with the collected data, I can now determine the rate of reaction. Once I have all my kinetics data, I can now perform computational analysis on them. With the collected data, I can now graph something called a Hammett plot. This is what an example plot will look like, with my rate of reaction on the y-axis denoted by K, and on the x-axis are the sigma pair values of each of the substituent. The Hammett plot describes the relationship between the rates of reaction and substituent effect. The graph could be linear and adopt a positive slope, a negative slope, no slope, or even changing slope throughout the reaction. The magnitude and shape of the slope can provide mechanistic information about the reaction. To conclude, I would like to emphasize the importance and relevance of studying reactions that prepare building blocks such as azetidines, as they can be applied to synthesis of essential pharmaceutical drugs and life-saving drugs. Lastly, special thanks to my supervisor, Dr. Dirksen, my mentor, Duncan, and everyone in the Dirksen group. Thank you.